Good morning, students. Now, today we are going to study about the characteristics features which are required in a cloning vector. Actually, in the last class, we were learning about the cloning vector and we studied the two cloning vectors, that are the plasmid and bacteriophages. Now, we will discuss today that what should be the features, what are the characteristics required in a cloning vector. First of all, in a cloning vector, there must be an origin of replication. So there must be an origin of replication, so that if any alien piece of DNA, when we linked with any plasmid or any vector, it helps to replicate. So there must be a origin of replication, so that when an alien piece of DNA or foreign DNA is linked with the cloning vector or linked with that vector, it helps in replication. So there must be an origin of replication. But the question is, what is origin of replication? See, in the DNA, there is the specific sequence of bases, sequence of nucleotides found, that is called the origin of replication. So if any alien piece of DNA is linked with this sequence, it can be made to replicate within the host cells. That is so there must be an origin of replication. One thing also we should know, this origin of replication also responsible to control the copy number of the desired gene or linked gene or any alien piece of DNA in the host cell. This origin of replication is also responsible to control the copy number of the linked DNA or desired gene. So we should select such kind of vector whose origin of replication support high copy number. We should select such kind of vector whose origin of replication support high copy number. So, first feature that it is required in any cloning vector is there must be a origin of replication. Origin of replication means this is the sequence of DNA, specific sequence of bases that initiate the replication, and when a piece of DNA is linked to this sequence, it can be made to replicate or multiply in the host cells. And also we learned that this origin of replication is also responsible to control the copy number of that linked DNA. So we should select such kind of cloning vector whose origin of replication support high copy number. So this is the one of the characteristics features that required in an cloning vector. Now we will discuss the second property or characteristics or features which is required in a cloning vector. So there must be a selectable marker. In the cloning vector, there must be a selectable marker. Question is, what is selectable marker and why there should be a selectable marker? Actually, in the cloning vector, if there is the selectable marker, it help to identify that which one is the recombinant, which are the non-recombinants. You can say that the which bacteria are the transformants and which are non-transformants, because in this biotechnology or recombinant DNA technology, what we do, first we take out a desired gene from any organism, then it is linked with the vector like the plasmid, that is called the now recombinant DNA, and that recombinant DNA is introduced into a bacteria. So actually this process occurs in culture medium. We do not introduce this recombinant DNA in one bacteria. Bacteria found in the culture medium, and there are the thousands of bacteria. We add the thousands of recombinant DNA in that culture medium. So in that condition, this recombinant DNA may enter in some bacteria, may not in the other bacteria. So in that culture medium, there must be some transformants. There may be some non-transformant. So we require only transformants having the desired gene for replication and for the large scale production of any industrial product. So there must be a selectable marker in the vector that helps to identify and eliminate the non-transformants from transformant and selectively allow the growth of the transformant, because we require only transformants for the large scale production of any industrial product, not non-transformant. So there must be a selectable marker. For example, suppose a cloning vector having ampicillin resistance gene, okay, ampicillin resistance gene. And now in that vector we have now linked an alien piece of DNA. And then this recombinant DNA is now allowed to enter or introduce into the bacteria. This process is called transformation. After this transformation, now we grow that bacteria in a culture medium having ampicillin. So when we grow that culture of bacteria in a medium having the ampicillin, in that condition the transformant will survive, because in the transformant there is the ampicillin resistance gene, but the non-transformant will die. So it can be selected, because if there in the cloning vector, suppose there is the ampicillin resistance gene, and in that vector we have linked an alien piece of DNA and then introduced into the culture of bacteria. So okay, in that culture there may be some transformant, there may be non-transformant. So what we do? Now we allow that bacteria to grow in a culture medium having the ampicillin medicine, ampicillin. So the bacteria having the ampicillin resistance genes are the transformant. They will survive, they will grow. But the non-transformants, they have no ampicillin resistance gene, they will die. So easily it can be identified that these are the transformants, these are not transformant. So there must be a selectable marker present in the cloning vector that helps to identify and eliminate the non-transformants from the transformants, because we require only transformant in this biotechnology for the large scale production of any product. What is transformation? Just now I said, suppose, now you can see this diagram also. Suppose this is the vector DNA or cloning vector, and now this is the foreign DNA or foreign DNA. First of all, what we do? We use the same restriction enzyme to cut these two, both DNA, vector DNA and the foreign DNA, okay? Same restriction enzyme. There are the many restriction enzyme. We use the same restriction enzyme to cut these two DNA. Now after that, now it cut this restriction enzyme, cut this vector DNA also, this foreign DNA also, and after that we add the ligase enzyme. You know that ligase enzyme helps to add the two DNA. So now this foreign DNA, or suppose this is the desired DNA, this is the piece of DNA, desired gene. Now we want to add or ligate this desired gene in this cloning vector, both together. Now we add the ligase enzyme. It now joins or link this DNA into this vector DNA. So this is the now recombinant DNA. This one is the recombinant DNA. You can see that this one is now recombinant DNA. This is the recombinant DNA. So there is the desired gene, and now after that this recombinant DNA is introduced into the bacteria, and when this recombinant DNA enter into the bacteria, this is the bacteria, this is now called the transformant bacteria. But if suppose in the bacteria, in the culture medium, there are the thousands of bacteria, and we add thousands of this recombinant DNA also in that culture medium, but in some bacteria it may enter, in some may not. So how to identify in this culture medium that which bacteria are the transformant and which are the non-transformant? So if there is suppose a selectable marker, suppose there is the ampicillin containing gene in this cloning vector, ampicillin resistance gene. So what we do, this transformant or after transformation now allow this bacteria to grow in a culture medium having the ampicillin. So the bacteria having the ampicillin resistant gene or the bacteria which are transformant, they will grow. They will grow because they have the ampicillin resistance gene. But the such bacteria having no this recombination or no transformation, non-transformants cannot grow in the medium having the ampicillin because they have no resistance against the ampicillin. So they will die. So there must be a selectable marker in the cloning vector. Okay, so this is the process. Now you can understand. Suppose this is the vector DNA, this is the foreign DNA, and we want to take a gene from this foreign DNA and add in this vector DNA. We use the same restriction enzyme to cut this both vector and the foreign DNA, because if we use the same restriction enzyme, it leaves the sticky end. There will be the sticky end. There is also a sticky end, and this sticky end helps to join this foreign DNA with the vector DNA. So to leave the sticky end at the end, and this is the end, this is the end. Okay, we use the same restriction enzyme to cut this both vector and the foreign DNA, so that it can be easily ligated, because it leaves the sticky end at the end. And if we add this foreign DNA with this vector DNA, and also we add the ligase enzyme, this ligase enzyme helps to join this foreign DNA or the desired gene with this vector DNA, and now it is called the recombinant DNA. After that, because in the biotechnology it requires the large scale production of the product, so this recombinant DNA also require in large number. We make the large number of recombinant DNA, and there is not a single bacteria. We do not introduce this recombinant DNA in single bacteria. The bacteria, we grow this bacteria in the culture medium. There is the thousands of the bacteria, and we add this recombinant DNA in that culture medium. So this recombinant DNA may enter into some bacteria, may not in other bacteria. So those bacteria, if this recombinant DNA enter or introduce, that bacteria are called the transformants, and the others are the non-transformants. So how to identify? Because in biotechnology we requires the growth of only transformant to produce the desired product, not the non-transformant. Okay, so there must be a selectable marker that helps to identify that which bacteria are the transformant and which are the non-transformant. Okay, thank you. Now we will discuss in the next.